In this video, we are going to discuss about the loop-mediated isothermal amplification based detection of target genes. So in this video, we are going to first discuss about the theory, then we are going to see how to perform the experiment in order to detect the target gene. Loop-mediated isothermal amplification is one of the isothermal nucleic acid amplification method. In isothermal nucleic acid amplification method, there are three prominent methods are used in the research field. The first one is the lamp, second one is RPA, third one is RCA. So the isothermal amplification, from the name itself we know that the amplification of nucleic acid or the target gene happens at the constant temperature. So in comparison to PCR or real-time PCR, where there is a different temperature is needed, but here in isothermal amplification method only you need one particular temperature to amplify the target gene. That is the advantage of using isothermal amplification. So in this, lamp is the most predominantly used method, which was invented in 2000 by a group of researchers in Japan. So isothermal lamp is amplifying the target gene at 60 or 65 degrees Celsius. So the second isothermal amplification method is called recombinant polymerase amplification, simply we call RPA. It amplifies the target gene at 37 or 42 degrees Celsius. The third method is rolling circle amplification, which is amplified at 30 or 37. So in this video, we are going to see only about the lamp. So this is the overall picture about how to do the lamp reaction. So the lamp reaction, basically we are going to use many number of primers. In contrast to PCR, where we use only 2 primers, here we are going to use 4 to 6 primers. So these 4 to 6 primers is able to amplify gene, target gene, in the formation of loop, as you see in the picture. So you can see the formation of looping structure. That's why the name comes. So at the end of the amplification, you get a fragment of DNA of various size, because the target gene joined one over another at each cycle. So that's why you will get a various size product. So if you see how to analyze or visualize this amplified product, there are different methods. The most common method is gel. You can use agarose gel. Normally we use 1.5 to 2% agarose gel in order to visualize, because the range of amplified product is vary from 50 to maximum to any number size. So we have to use higher percentage of agarose gel. Second one is you can use a calcium calcium based fluorescent method, or you can use a HMB based method. So otherwise you can use a cyber green, which is a fluorescent dye, which you can use a UV lamp in order to detect the amplified product. So there are various methods that we can use to see the visualized product. So this makes this product is most viable for the on-field detection method of the pathogens. So though this is amplified in a isothermal condition, most of the time it is only used for the detection purpose. LAMP method uses BST DNA polymerase. So the BST DNA polymerase optimum temperature of the enzyme is 60 to 65 degrees Celsius. So that's why the amplification is always happening in isothermal. So this enzyme has a strand displacement activity. That's why we don't need different temperature in order to do unwinding and extension of the primers. So strand displacement activity means the incoming synthesizing strand will replace the existing strands, even though in the duplex formation. That's why we don't need a unwinding step in this lamp. The amplification of BST polymerase is very, very exponential. That's why even within 30 to 35 minutes, so you can able to get the amplified product. In contrary to the PCR, where you need 2.5 hours. That makes this lamp is more potential candidate for point of care devices. So if you can see here, So this is the structure, explain how the primers are actually binding to the target gene. So here there is only 4 primers is shown. So 4 to 6 primers you can use. These 4 primers is very mandatory. These 4 primers, there are 2 outer primers which is called FIP and BIP. There are 2 inner primer, F3 and B3. F3 and B3 primers are called outer primer, which is similar to our PCR primers. Whereas the inner primer is very specific to the lamp, which you can see in one inner primer, normally the bases is 440 to 45 base pairs. So the one inner primer can target 2 region of the gene, target gene. So totally there are 6 region is targeted by these 4 primers. So this makes this very specific in comparison to PCR or conventional PCR method. So if you see how this product, amplified product, is visualized in the real experiment. So the left hand side, the figure 3 shows that it shows the agarose gel. So in this we have performed different times starting from 60 minutes to 15 minutes. You can see the amplification even slightly visible in 30 minutes, where it is very predominantly visible in 45 and 60 minutes. Always you can see the ladder-like structure like you see in the picture. So as I already mentioned, there is a different size amplified product you can get at the end of each cycle. That's why you will get the ladder-like structure. Whereas figure 4 explain how the calorimetric method we can detect. So in calorimetric method, whenever there is a, we are added HNB, in the presence of HNB, whenever there is a amplification, there is a color change from purple to deep blue. That's what you can see. And today's this particular lab, we are going to see how to perform the experiment using lamp. So this particular method is shown about the lamp mixture. That is, typically you have 25 microliter lamp mixture. So this is the mixture that we are going to use in today's experiment.